Yes, preparing to live stream. Confirm, please, when you have it.
Yeah.
Okay.
You're good.
And I'm going to make Peter, I still will make you a co-host. Peter, if you want to share your screen. There we go. Confirm, please, when you have it. And let me shut down. Okay. So good morning, everyone. I apologize. There's always something, no matter how many. I mean, we've done it 100 times, and that's not a random number. Peter, you get the award for being the 100th presenter of Fine over the course of 3 years. So even after 100 times trying this, something happens. If it is not going live, it's something else. So before introducing our speaker today, and to save a few minutes, I will not share my screen. Let me remind you that, or maybe I'll do, because I want to show it. Yes, let me show you quickly. So you may remember, it was a fantastic talk we had last week. Mark Harrison presented on molecular signatures of longevity in eusocial animals. For those of us, definitely for those of us working with mammals, learning about these taxa not showing the relationship between longevity and aging is very, very interesting. Next week, for those of you who need to go after Peter finishes, we have Josh Firth from the Department of Biology at Oxford University. He will be presenting a talk on the behavioral ecology of contagions, pandemics, passerines, and permuted populations. But now what we want to do is we want to welcome our speaker, who is Peter Buston from Boston University in the US. Peter will be talking about social evolution in anemonefishes. And Peter got his first degree at Oxford University in the UK. From there, he went to Cornell University in the US in the Department of Neurobiology and Behavior. He completed a couple of postdoctoral fellowships. The first one in Santa Barbara, California. I guess he must have been tired of the cold in Cornell. So he went down to the south and he stayed for a couple of years at the National Center for Ecological Analysis and Synthesis. He liked the heat, so he went from there to the Estación Biológica Doñana in Spain. And after completing that second postdoc, he joined the faculty at Boston University, where he's been ever since. His system has been for a long time now working with fish. And today he will be talking primarily about one of the two big projects he has. So you've already seen the title, you have seen the abstract, you know that he does a lot of work on social evolution, focusing on an understudied system, fish, and in oceans. But the other topic that he will not be discussing today, but I invite you to check, is that he does a lot of work on population connectivity and dispersal. Again, focusing on fish, focusing on coral reefs, and trying to understand meta-population dynamics, which he was telling me right before is a little bit of a black box. I was telling him that I think dispersal continues to be a black box, not only in the oceans. For most taxa, I think we have an awful lot to learn about dispersal. Just to give us, I'm not going to go there or it'll take too long. So with that, I think that I covered what I wanted to say about Peter, who is gonna, I'm sure he's going, well, he's done an awful lot of outreach and broader impact. So I suspect we're in for a wonderful talk that not only is going to present some of the very, very solid science he's done, but probably with some connections and references to the fantastic training and outreach that he's done in the various countries where he works. Peter, if you would like to share your screen now and take it from here, and welcome to the Fine series, the 100th presentation of this series. Congratulations to all of us.
Thanks very much, Eduardo. It's lovely to be here. Thanks for inviting me to present for this 100th occasion. For those of you who just turned up, we were having a little conversation earlier. I dropped a couple of popular science articles in American Scientist in the chat, one in English, one in Spanish, for those of you who are interested, and they might be nice for sort of undergraduate classes and things like that. So today I'm going to talk about social evolution in clownfish. So Animal societies in which some individuals forego their own reproduction and help others to reproduce are one of the most remarkable products of evolution. And these can be ants, bees, wasps, termites, social cockroaches, shrimp, naked mole rats, bee eaters, fishes like the cichlids. We all have our favorite sort of cooperative breeding or eusocial taxa. And these have been of interest to evolutionary biologists ever since Darwin. Right there in the middle of the origin of species, he identifies one special difficulty, which at first to him appeared insuperable and actually fatal to his whole theory. And he was alluding to the neuters or sterile females of insect communities. To frame Darwin's sort of special difficulty in modern terms, when you have these non-breeding phenotypes, it's not immediately apparent how they get their genes into the next generation. And so it's not immediately apparent how they persist for more than one generation. We move on to the 1960s, and there is two sort of major hypotheses emerging that help to explain this. So first of all, W.D. Hamilton proposed that there are two ways, or noted that there are two ways for individuals to pass on their genes. They can either do so directly by producing their own offspring, or they can do so indirectly by enhancing the offspring production of relatives. And so it could be that these non-breeders are helping relatives and thus passing on their genes in that way. A second sort of complementary hypothesis that emerged is the ecological constraints hypothesis, which pointed out that there are two alternative options available to individuals. Either they can stay on their natal territory as non-breeders, or they can disperse from the natal territory to breed elsewhere. And when you take these two hypotheses together, you can come up with an evolutionary framework that can explain these non-breeding strategies. Okay, so if you take kin selection and ecological constraints, they can help to explain why some individuals forego their own reproduction and help others to reproduce. So this inequality is one formulation of Hamilton's inequality. So if you look at the X terms, Xi or Xj, that's the non-breeder's fitness associated with its actions. It's here and here. Yi or Yj is the breeder's fitness associated with the non-breeder's actions here. R is the probability of sharing an allele identical by descent. And then the I terms represent the payoff associated with staying and potentially helping. And the J terms represent the payoff associated with leaving. Okay, so in essence, non-breeders have a 0 here, but they may have some contribution to the next generation if R is positive and if they enhance the fitness of the breeders. And staying and helping can be favored by selection if there's some ecological constraint such that this term on the right is small. Okay. But those aren't the only two hypotheses. We can think of a third way for individuals to get their genes into the next generation. And this is that they can do it directly by producing their own offspring in the future. And so sort of idea here is that when we go and we look in the field as a society, what we're seeing is a particular snapshot in time. And these non-breeders might not be non-breeders forever, but they may inherit the territories in the future and pass on their genes as breeders that way. There's also a third option available to individuals, and this is that they can contest to breed on the natal territory. Okay, so often this isn't talked about so much, but this idea that individuals can contest to breed is something that they can do, just like they can disperse to breed elsewhere. And so to understand non-breeding strategies, we need to understand why the payoff from contesting is also small. So you could put these ideas together in the same way as we did for kin selection and ecological constraints. So future selection and social constraints can help to explain why some individuals forego their own reproduction and help others to reproduce. X is the non-breeder's fitness associated with its actions in the present. Z or Z is the non-breeder's fitness associated with its actions in the future. F is the probability of realizing those benefits in the future. And the I terms represent the payoff associated with staying and helping, and the K terms represent the payoff associated with contesting. And so essentially, non-breeders can be favored to stay and help, even if they're not getting anything. If this is 0, so long as there's some non-zero probability of territory inheritance, and so long as the payoff associated with contesting is really small. Okay, then it can pay for them to wait to inherit the territory and then breed on their own in the future. So this is all well and good, but there are some limitations on the framework when I was getting started as a PhD student. The framework had very much been developed and informed by studies of birds, mammals, eusocial insects, and its generality was perhaps unclear beyond these taxa. So myself and colleagues set out to test the robustness of this evolutionary framework by investigating a new taxon in a new environment, which might impose different constraints. Okay. And I should say, in the 25 years since I've started, the taxonomic scope of these kinds of studies has increased markedly. Okay, so this led me to study clownfish societies. This is probably my favorite fish in the world. It's the clown anemonefish or orange anemonefish, Amphiprion percula. It's a spectacular looking fish. And it lives in groups, and these groups are found in close association with sea anemones, like this one here. So each group is composed of a breeding pair and some non-breeders. So this is the breeding pair. This is a non-breeder. And within each group, there's a size-based dominance hierarchy. So this is the female, she's the largest, always. The male is always the second largest here. And the non-breeders get progressively smaller. Sort of an interesting side point is that clownfish are sequential hermaphrodites. So if the female of this group dies, the male will change sex and become female. And also nicely, breeding occurs year-round on a lunar cycle. And so unlike if you study birds or something like that, if I'm studying clownfish, I can get 12, 13 breeding seasons into a year if I spend a year in the field. So what I want to talk to you about today, just had the introduction, and I want to talk about testing 4 hypotheses for why these non-breeders tolerate their situation. We'll talk about kin selection, future selection or territory inheritance, ecological constraints, social constraints, and then I'll wrap up and I'll also tell you about future directions. So first off, kin selection. So the hypothesis makes 2 critical predictions: that non-breeders enhance the fitness of breeders such that yi is greater than yj, and non-breeders are closely related to the breeders, so ri is greater than 0. Okay. So to test prediction 1, we conducted a year-long study of 97 groups of Amphiprion percula in Madang Lagoon in New Guinea. This is me during my PhD work many years ago. So I would basically map out 97 groups. Very nicely, these groups don't move. Their anemones are static, so I can go back and find the same groups every day. And I monitored the survival, growth, and reproduction of 71 groups, and I conducted an experimental removal of non-breeders from 14 groups. Okay, you have to do this experimental removal in this particular system because female size is correlated with group size, is correlated with anemone size. And so you've got this triangle of correlates that makes it really difficult to infer causality. And the only way to infer the effect of non-breeders is to remove them experimentally. So after a year in the field, an enormous amount of work boiled down to this one figure. We've got the number of eggs hatched by breeding pairs per lunar month here on the x-axis, on the y-axis, sorry. And on the y-axis, we've got control groups and manipulated groups. And in the white, we have their reproductive success prior to the removal of non-breeders. And in gray, we have post-removal of non-breeders. So obviously, the control groups didn't have their non-breeders removed. So this is just a seasonal control. Okay. What I want you to take home from this figure is that basically removing non-breeders has absolutely no effect on the reproductive success of the breeders. And it turns out it doesn't affect the survival, growth, or the egg production of the breeders. Okay. To test prediction 2 of the kin selection hypothesis, we developed 7 polymorphic microsatellite loci and genotyped 32 individuals from 9 groups. And we were able to show that non-breeders are not closely related to the breeding pair. Okay, so this is relatedness on the x-axis. This is the average relatedness within groups, which is squarely centered on 0. And this is the pairwise relatedness within groups, which is also squarely centered on 0, unsurprisingly. Okay, so what does this mean? It tells us that in the clownfish, non-breeders do not enhance the fitness of the breeders, and non-breeders are not closely related to the breeders. And this is interesting because it means that kin selection does not explain the evolution of non-breeding strategies in the clown anemone fish. And this is likely true for most coral reef fishes because dispersing larvae break up kin associations in the sea. And so non-breeding coral reef fishes that occur in a few taxa must be contributing genetically to the next generation in some other way. That brings us to hypothesis 2, the idea of future selection or territory inheritance. And there's just one critical prediction here, and that's that the non-breeders will inherit the territory on the death of the breeders. So this Fi is greater than 0, such that the non-breeders get to breed in the future. So to test this prediction, again, part of the same year-long study of 97 groups of clownfish in Madang Lagoon in New Guinea, we monitored naturally occurring breeding vacancies in 57 groups, and we experimentally created breeding vacancies in 16 groups by removing the male to see who would inherit that position. And this is probably the cleanest data set I've ever produced, but this is the pattern of territory inheritance. So here you see we've got the fate of the breeding vacancies, whether the non-resident was usurped, a non-resident usurped the vacancy, whether a resident filled the vacancy, whether it was the largest non-resident, and whether the new pair successfully bred. And so for natural vacancies, there were 9 of them. None of them were usurped by an individual from another anemone. In 7 out of the 9 cases, a resident filled the vacancy. In the case where there were 2 non-breeders, it was the largest that filled the vacancy. And in 5 of these cases, the newly established breeding pair bred before the end of the study. The other vacancies were filled by larvae settling into the anemones, not by sort of reef residents moving. And the reason that some of these hadn't bred by the end of the study is because the naturally occurring vacancies occurred close to the end of the study. If we look at the experimental vacancies, it's sort of the perfect data set that you could only wish for. Non-residents usurped the vacancy in 0 cases. In all cases, it was a resident non-breeder that filled the vacancy. In the 7 cases where there was more than 1 non-breeder, it was the largest non-breeder that filled the vacancy. And in all cases, the new pair successfully bred. So in the clownfish, non-breeders do stand to inherit the territory in the future. And so future selection, as I like to call it, helps to explain the evolution of these non-breeding strategies in the clown anemone fish. So future benefits are necessary, but they're not sufficient to explain the behavior of non-breeders. A complete understanding requires that we also explain why they don't disperse to breed elsewhere. Why is it that they're staying in these groups for a long period of time, waiting to breed? And this question becomes more compelling when you understand how long clownfish live and how long they queue for. So clownfish have an extraordinary life expectancy, very much like naked mole rats. Females can be over 30 years old. And this means that, and they have a low mortality rate. And this means that individuals can be queuing as non-breeders for 5, 10, 15, or 20 years. Okay, but they always stay in the group. They never disperse to breed elsewhere. And we want to understand why that's the case. So that brings us to the ecological constraints hypothesis. Hypothesis makes 2 critical predictions. Individuals will stay as non-breeders when there is some ecological constraint. So the right-hand side of this inequality is particularly small. It also makes the critical prediction that the likelihood of individuals dispersing will increase if you relax that ecological constraint. So what might the ecological constraint be? Well, there's two types of ecological constraint for clownfish. First of all, the anemone habitat is saturated on these tropical reefs. Every single giant sea anemone is occupied by clownfish. And secondly, movement between anemones is extremely risky. So the anemones provide protection from predators. Clownfish are brightly colored and they're poor swimmers. And if you take them out of their anemone, as Vic Mariscal did in the 1970s, you'll find they're quickly preyed upon. Okay, so the ecological constraints for clownfish are a risk of movement, and even if you do successfully move and find another anemone, that anemone is going to be occupied by territorial residents. Okay, so the first tests of the ecological constraints hypothesis we didn't move any anemones. We created habitat vacancies by removing non-breeders and males in the experiments that I've previously described to you. When we did this, no individuals dispersed between anemones, even when habitat vacancies were created in this manner. Okay, so one possible explanation for that is that the non-breeders just didn't have information on the vacancies. So we've more recently done a sort of better experimental test. And this was led by Rebecca Branconi, who's now based back in Florence in Italy. What she did was she placed alternative anemones at 0.5 meters from focal anemones, and she looked to see if the non-breeder from the focal anemone moved to take up a breeding position in one of these alternative anemones. She provided two types of alternative anemones, one that had a female so that this individual could move and become a breeder with it, and the other option was an empty anemone. So slightly different options for these non-breeders. Her intent was to provide that at 0.5 meters and 5 meters to see the effect of distance on the dispersal of the non-breeders. But as you can see here, here are counts of dispersal on the y-axis. Here's the weaker ecological constraint. This is the closer anemone at 0.5 meters. And when we presented these anemones with breeding vacancies at 0.5 meters, the non-breeders never left their home anemone. Okay. So there's two potential explanations for this. One is that they simply don't have movement in their behavioral repertoire, much like a bunch of sort of eusocial insects. Another possibility is that there's some benefit of being in a good anemone with sort of known social relationships, such that they don't leave. So to tease apart these alternatives, we redesigned the experiment. And Rebecca placed the alternative anemones, again, next to the focal anemone. But this time she took the non-breeder out of the focal anemone and moved it to the alternative and looked to see if it would return home. Okay, and again, she did this at 0.5 meters and at 5 meters. And these results are quite nice. When the alternative anemone and the non-breeder were placed at 0.5 meters, a good number of them returned home. So you can see sort of more than half of them returned home. When we placed the non-breeder at 5 meters, none of them returned home from 5 meters. So conclusions here are that found anemone fish individuals stay as non-breeders because there are harsh ecological constraints. It's dangerous to move, all the habitat's taken. But individuals will disperse to breed elsewhere when that ecological constraint is relaxed. So it is in their behavioral repertoire. Okay, so this is nice because it's like saying, well, kin selection, which is a big driver of social evolution in terrestrial systems, isn't operating here, but ecological constraints are operating here. So future benefits, the territory inheritance in conjunction with ecological constraints can explain why non-breeders wait rather than disperse, but they don't explain why individuals wait rather than contest. And again, I remind you that these individuals are waiting 10, 15, 20 years to inherit breeding positions. If I were in a queue that were taking, and it was going to take me 10, 15, or 20 years to get to the front of the queue, I might consider fighting with the people in front of me to get to the front. But clownfish don't. And this brings us to the topic of social constraints. So you can think of this just in the same way as you think of the ecological constraints. And there are two critical predictions. Individuals will wait as non-breeders when there is some social constraint such that the right-hand side of the inequality is made small, but also that the likelihood of individuals contesting will be increased when that social constraint is disrupted, just like the likelihood of individuals dispersing will increase when the ecological constraint is disrupted. So what might the social constraints be in clownfish? Well, it turns out that if you measure all of the fish in the population, you find that subordinates tend to be 80% of the size of their immediate dominant. Okay, so here we see, this is just a histogram. So we have relative frequency on the y-axis. We have the size ratio between the subordinate and its immediate dominant on the x-axis. So that's the size ratio of, that's the size of rank 2 over the size of rank 1, or the size of rank 3 over the size of rank 2. The purple distribution here is the naturally occurring distribution of size ratios. And the yellow distribution is the distribution you get if you just randomized the individuals in their groups. Okay, and there's a big difference between these two distributions. First of all, you see that under natural conditions, there's an overabundance of size ratios around 0.8, 80%. And also under natural conditions, there's very few size ratios close to 1, 0.0. And it turns out, which we show in this 2003 paper, subordinates are effectively growing to 80% of the size and then of their immediate dominant, and then they're stopping. And at that size, their probability of winning a contest is close to 0. It's effectively 0. So this is the social constraint for subordinates. At 80% of the size of their immediate dominant, they can't win a fight. And so they can't basically contest. So what happens when we relax this social constraint? So Rebecca, again, did a very nice experiment where she took this focal group, she removed the resident rank 3, and she introduced an individual either a few millimeters bigger, which means that their social constraint is relaxed because it now has a better chance of winning a fight, or a few millimeters smaller, which means that the social constraint is stronger because it has less chance of winning a fight. So she has counts of contests and evictions here on the y-axis. Contests initiated by the subordinate often result in eviction. And here on the x-axis, we have the weaker social constraint or the stronger social constraint. What you can see is that larger non-breeders contested and were evicted in 12 out of 16 cases, and smaller non-breeders only contested in 3 out of 16 cases. So it turns out that if you relax this social constraint by giving the non-breeder a growth spurt, it will contest for these breeding positions. So in the clownfish, individuals will wait as non-breeders because there are strong social constraints. But individuals will contest for breeding positions when you relax that social constraint. So future benefits and ecological constraints can explain why non-breeders wait rather than disperse to breed elsewhere. And future constraints and future benefits and social constraints can explain why non-breeders wait rather than contest. Okay. So to wrap up this part of the talk, we started with Darwin's sort of evolutionary puzzle, this one special difficulty, alluding to the neuters or sterile females of insect communities or cooperative breeding groups. And it might be that we've at least solved this puzzle for clownfish. So 4 key concepts, so kin selection, future selection, ecological constraints, and social constraints seem to provide quite a complete understanding of the diversity of non-breeding and helping strategies in complex societies, including clownfish. We are left with another evolutionary puzzle, though, and that is, why do breeders tolerate non-breeders? Okay, we have these large, more powerful individuals who apparently get no benefit from the presence of non-breeders. And so it's reasonable to ask, why on earth do they tolerate them? And this leads me to the future directions that we're interested in. So these future directions are being explored in collaboration with Dr. Teresa Ruger. She was a Marie Curie postdoc with me. She's now a lecturer at the University of Newcastle. in the Center for Behavior and Evolution. And she is sort of spearheading answering this next question of why the breeders tolerate the non-breeders. So you can use the sort of same evolutionary framework. You're just considering different options. So kin selection and future selection can help to explain why breeders might tolerate non-breeders rather than evict them from the group. Okay, so what we've got here is the payoff to the breeders if they tolerate the non-breeders, and this is the payoff to the breeders if they evict the breeders. And they'll tolerate the breeders rather than evict them if the left-hand side is greater than the right-hand side. So the first question you can ask, the first hypothesis is the null, that breeders actually can't evict the non-breeders. But we've shown through multiple experiments that breeders can and do evict non-breeders if they get too large or attempt to reproduce. So the paper I just described by Rebecca Branconi showed that if we manipulate the size of the non-breeders, the breeders will evict them. This nice paper by Teresa in Proceedings in 2018 showed that if we remove this rank 3 individual, we introduce a size-matched rank 3 from elsewhere, or we introduce a size-matched male from elsewhere, then the males get evicted, but the rank 3s do not. Okay, so this is really interesting. The breeders, when they're presented with unfamiliar individuals of a particular size, they can tell whether that unfamiliar individual is reproductively active or not. And if it's reproductively active, they evict it. So we falsified the null hypothesis. Breeders can definitely evict the non-breeders. Okay. We've also falsified hypothesis 1 that the breeders accrue some sort of direct genetic benefits. I showed from my PhD work that basically breeders get nothing in terms of survival, growth, or reproduction from the presence of non-breeders, at least on sort of short timescales. The next hypothesis is that breeders accrue indirect genetic benefits from the presence of non-breeders. And this might work, as pointed out by Hannah Coco and Rufus Johnstone in their queuing models, if there's no opportunity for non-breeders to breed elsewhere, but they go on to inherit the territory on the death of the breeders, then it can pay sometimes to tolerate your relatives within the territory because they get to inherit your territory. But we've shown back in 2007 that non-breeders are not closely related to the breeding pair. But we did this using a small panel of microsatellites and a relatively small number of individuals. And things have advanced quite a lot, and a few studies, including one by Teresa on coral gobies, have started to show that there can be some subtle patterns of relatedness among individuals within groups. And so it's quite possible, we think, that the non-breeders are distantly related to the breeding pair. Perhaps they are great-great-grandchildren or second or third cousins who go on to inherit the territory. And while these coefficients of relatedness of around 0.025 or something like that are tiny, they can tip the balance in the inequality such that it may pay the breeders to tolerate the non-breeders. So we're exploring this now in clownfish. Another reason that breeders might tolerate these non-breeders is they serve as rapid mate replacements. And this was the original hypothesis proposed. The idea is that in the event that one of the breeders is widowed, by having a non-breeder there, they can recommence breeding more rapidly. I tested this in my early work and showed that there's a relatively trivial benefit that accrues from non-breeders being rapid mate replacements. They don't really reduce the mean time to recommence breeding. What Teresa and I are interested in now is that they may dramatically reduce the variance in time to recommence breeding. And that's because if a non-breeder isn't present and a breeder is widowed, while most of the time a non-breeder turns up, new larva settles to anemone and it can breed quite quickly, there are rare occasions when in particular points in space or points in time where a breeder may have to wait a year or even 2 years for a larva to turn up. So what we think is that actually sort of a bet hedging idea is that by having these non-breeders around, breeders may reduce the variance in their reproductive success because they reduce the variance in the time to recommence breeding. And so we're looking into that now. Finally, and I think most excitingly, is the idea that breeders might accrue mutualist-mediated benefits from the presence of non-breeders. So non-breeders might enhance the fitness of the anemone. They could enhance its health, or they can enhance its size. And a large, healthy anemone may benefit the breeders. Okay, and there's some correlational evidence for this in this species, and some experimental evidence for this side of the linkage in other anemonefish pairings. And this is really interesting because this is like, makes anemonefish a vertebrate example of fungus-growing ants or acacia ants. They are actively engaged in this mutualistic relationship with sea anemones, and they're dependent upon the sea anemone for their own lifetime reproductive success. And so one reason why the breeders might tolerate the non-breeders, just like in those ant societies, is because they're enhancing the success of the mutualist partner. And so we're looking into this. Now we have a long-term 2-year experiment plan to tackle this question. So where are we right now in this question of why do breeders tolerate non-breeders? We've rejected the null. Breeders can definitely evict non-breeders. We've rejected the obvious. We've shown that breeders don't accrue direct genetic benefits in the present. We're investigating whether breeders accrue indirect genetic benefits. This is like with a low coefficient of relatedness, sort of weak kin-selected benefits. We're investigating whether breeders accrue hedging benefits because the presence of non-breeders reduces the variance in the time to recommence breeding. And we're investigating whether breeders can accrue mutualist-mediated benefits in a similar way to sort of fungus-growing ants or acacia ants. So stay tuned for that. But with that, I will wrap up, and I look forward to a conversation. Just some acknowledgements. So for permissions and logistical support, the government of New Guinea, the landowners of Riwo Village in Madang, where I did my PhD work, the Christensen Research Institute, Jai Zarban Resort, and my boat captain, John Mijau. And then more recently with Teresa, we've been working on the reefs that are owned by the landowners of Tamari and Kilu and Kimbi. There we get logistical support from Mahoney and Hadari Research Station and Walindi Plantation Resort. And we've had the same boat captain for many years, Nelson Sikatua. Also to my advisors, students, collaborators over the years. None of this happens without them. And then funding for this project has come from the National Science Foundation, from the Christensen Research Institute, from Mahoney and Hadari, and some other small funds. And with that, I will take any questions.
Thank you very much, Peter, for a very, very interesting presentation. Let me remind those of you who join us today that what we do now is we move to some time for questions and answers. That has really, it's becoming more and more of a tradition at FINE, and people keep telling us how much they value this period when we get to talk to each other and hear each other's comments, thoughts, contributions. The way we do it is I invite you to type a question mark in the chatting window, and I will be calling those who write a question mark in the order that you post them. We give priority to students and postdocs, our younger colleagues, and it's been a while, I think, that we haven't heard from our YouTube community. Lauren, if you have anyone there who wants to ask a question, please bring them up, because we want to hear from you as well. So with that, since I see lots of questions, we might as well get started. And the first question, please, in the spirit of continuing to build a community at FINE, 2 things. We usually open the Zoom 15, 20 minutes before the hour. So if you want to stop by, say hi, join us early, you're more than welcome. And now when you're going to ask a question, please tell us your affiliation, your name and affiliation. With that, I'll take the first question, which comes from Rag.
Hello, can you hear me?
Yes.
Yes.
I am Raghavendra Gadakar from the Indian Institute of Science. I study social wasps, which have many similarities but interesting differences from the system that you described. Thank you, Peter, for this very, very clear presentation. In fact, it was so clear that I think I can find one or two things that I think were missing. Usually it's very difficult because the presentation is very complicated. This time it was very, very nice. So two things I thought might have been missing. One is you told us if they don't leave because it's too risky, but if they stay and they're small, then they can't fight, they cannot contest. And so it may be that by the end of their life, they don't actually get to reproduce. So can we actually tell whether the risk of leaving is more than the risk of staying? The risk of staying being not being able to reproduce at all. Can we actually balance these two? That's the first one. And the other is...
So let me take that one first.
Yeah,
and then
sure.
go to the other one. So the answer is yes, we can. Because it's a queue and I've been able to estimate the mortality rates of all the ranks in the queue, you can then estimate the probability of ascending in rank. Okay? And so then you can estimate the probability of ascending in rank by queuing, and you can compare that to the probability of ascending in rank by dispersing to breed elsewhere, things like that. And so there's some really interesting dynamics in queues. It turns out that your probability of ascending in rank is higher at the back of the queue, even though your probability of inheriting the breeding situation is lower. And the reason for that is there's more individuals ahead of you to die. Okay, so at the back of the queue, they actually have quite a high probability of ascending in rank, sort of over 60%. And so that greatly outweighs their probability of surviving to breed elsewhere if they left. Okay, and it greatly outweighs their probability of surviving to breed if they tried to contest. Okay,
but so is the little increase in rank useful at all if they don't get to breed?
Well, going from number 6 to number 5 is the only way to get to number 4, and that's the only way to get to number 3, which is the only way to get to number 2. So every step is helpful, yes. It turns out that the lowest probability of increasing in rank by queuing, by waiting, occurs at rank 3, because you only have 2 individuals ahead of you to die. And what's really interesting, in the orange clownfish, we never see individuals leave. I think it's just too costly for them. It's just too risky. So even at rank 3, it pays for them to wait rather than leave or contest. There are some other anemone fishes which are a bit more mobile, other species. And interestingly there, that when you see movement between groups, it's often a rank 3 that moves to take a vacant male position nearby. So there's some really interesting interspecific comparisons to be done on these sort of queuing dynamics.
But can you actually tell what fraction of the individuals never get to reproduce before they die?
Yes, I could do the math to do that. Well, first of all, of the eggs that hatch, only 1 in 10,000 make it into an anemone. And then of individuals that make it into an anemone, it's a long time since I've done those numbers. I think it's 8% actually get to breed by queuing, something like that. But it's the only way.
My other question was, is there a cost of tolerating non-breeders? If not, why bother to evict them?
Well, so that's a great question. There's not only do the non-breeders provide no benefits in terms of survival, growth, or reproduction, but they apparently don't inflict any costs either, right? So the null hypothesis is that the non-breeders are effectively neutral, and there may be no benefit. And so what we're interested in and where we are right now is we haven't demonstrated any benefit. What we're interested in is there some subtle benefit that is getting the breeders to tolerate the non-breeders? And so we think that that might come from them being distantly related and going on to inherit the territory. We think it might come from them reducing the variance in wait times to recommence breeding. And we think it might come from the mutualist mediated benefits. It could be that all those hypotheses get falsified and that's fine. And then we can all be in agreement that the breeders tolerate the non-breeders because they're neutral, but they need testing. So
good. Thank you.
Thanks so much.
Thank you, Raj. Thank you, Peter. We're going to go to students who are joining us from South Africa. They are brave and they're in the field station. It's cold. Thanks for joining us. So, Siyabonga, I hope I'm pronouncing your first name well. If you would like to Post the question you have, introduce yourself, tell us where you are. I know, but maybe the audience doesn't. And what's your question? We cannot hear you. You're not muted, but we're not hearing you. No, still not. We have another. Why don't we do this? Let us know, or just give me a thumbs up when you think it's working. We'll take another student. We have another question from our student who was next on the line. So Jonathan, please, your question.
Hello, I'm Jonathan. I'm a PhD student in anthropology at Yale. Thank you for the talk. It's really fascinating. And I also really enjoyed the papers that you shared. So thanks for those too. And in the papers you mentioned, or you talked some about how the markings on the fish and the fish's ability to discriminate individuals from each other was important for their ability to evict and keep fish that they didn't want in their anemone out. And what confused me is I was unsure of why being able to discriminate individuals was so important for evictions. Like, why don't the clownfish don't indiscriminately evict any fish that matches the size proportion that demands eviction, basically?
That's I'm trying to think how best to answer that. I guess the answer is it probably just helps you keep track. I mean, so if individuals are moving around in the anemone, they're going under the edge of the anemone, you have to have some way to assess your own size, the individual size, your relative size. And having done that, if all fish look basically identical, which they do in some other anemone fishes, then having figured out who you want to evict, if you can't keep track of that individual because there's a school of identical looking fish, all of whom differ marginally in size, then I think it makes the problem harder. And so I think it probably pays both the subordinate to sort of indicate its identity such that it can say, I'm subordinate number or subordinate P, you know who I am, and you know that I'm cooperating in terms of size. So I think it's probably in the subordinate's best interest to be recognized, perhaps. Does that?
Yeah, that gets at it. Thank you.
Okay.
So we're going to try again. They're switching computers. So I'm calling you if you have a question, but I wasn't sure whether it was you or Siyabonga.
Yeah, it's me. Can you hear me now?
There's a lot of back. Try again. Now I think
okay. Yeah. So hello everyone. I'm Jun. Okay, I'm Jun Yu from the University of Strasbourg. I'm doing PhD on the personality of bush karoo rat in South Africa. So my question about the last question, like why the breeder tolerates the non-breeders. I just want to ask if it's possible because it's difficult to travel for them to travel between these groups. Could it be that the breeder tolerate non-breeders because they want them to be there like a backup, like if their partner dies, they can just take one from the non-breeders?
Yeah, so this is the original hypothesis that was proposed to explain why anemonefishes tolerate non-breeders. It was proposed by Fricker in the 1970s. And it's the idea that on the death of your mate, so if one of the breeders is widowed, you might recommence breeding more quickly if there's a non-breeder there. So I tested that experimentally as part of my PhD work, and having non-breeders around confers about a 2% relative fitness advantage versus not having non-breeders around in terms of mean fitness. Once you take into account like the number of non-breeders they're going to use and their sort of breeding tenure and things like that, which isn't a huge amount, but it can tip the balance in favor of tolerating these non-breeders. Teresa and myself think there's a much bigger benefit that comes in terms of non-breeders reducing the variance in the time to recommence breeding. And that's because on coral reefs, larval recruitment is sort of a stochastic process, and you're never guaranteed that there's going to be a larva arrive at the anemone if you're in a particular anemone at a particular place at a particular time. And we've seen cases where anemones exist with just one individual for over a year. Okay. And actually we've done some simulations now, and it turns out there's some sort of non-zero probability that you might wait 2 years or even longer for a non-breeder to arrive. And so what we think is that by tolerating these non-breeders, you're just avoiding these rare long wait times for a mate replacement. So
Yeah. Okay.
So slightly different from the original, but with the focus on the variance in the time to recommence breeding rather than the mean time to recommence breeding. Thanks.
Okay.
It's a good question.
Thank you.
I'm delighted to have so many students willing to ask questions. So I'm going to ask from my senior colleagues to be patient with me. We're going to go straight to Facundo Fernandez Duque, who I believe is the next student who would like to ask a question. Facundo.
Hi, I'm a graduate student in Allison Bell's lab and Mark Halbert's lab at UIUC. I'm looking at parental care stuff, but I was interested, do you happen to know, I don't know if you mentioned it, the survival probability of being evicted?
Yeah, it's effectively zero. So these evictions are really interesting. You end up with the residents basically driving a non-breeder or whoever's being evicted just outside of the periphery of the anemone. And then there'll be a standoff where the evictee is trying to get back into the anemone, but is outside of the safety of the anemone's tentacles. And they'll have a standoff that can last a day, 2 days, 3 days until this individual disappears. And basically it's until it probably gets eaten. In all the experiments we've done all over the years, there is only one case where I've seen an evicted individual basically give up and make it to another anemone. And the only reason it was able to do that is in the process of doing these eviction experiments, I had removed an individual from an adjacent anemone that was basically the same size of this individual that got evicted. So it moved down the reef and it just happened to encounter a gap of exactly the right size to slot into that anemone. Under natural conditions, that would almost never happen because it would have arrived at the new anemone, the size hierarchy would be filled, and those individuals would also have evicted it, right? So it would basically bounce around anemones, but ultimately it's going to get eaten.
Okay, thank you.
Thanks.
So let's hear next from Eugenia, and then we go to Siyabonga, and then to Adriana. Eugenia.
Okay, hello. I work at the Public Veterinary Service in Rome. I'm a biologist, and I work on indicators of stress in the dogs in the shelter and on reproductive strategy in cats and dogs, free-ranging cats and dogs. Thank you, Peter, for the talk. It was fascinating, really. And I have just 3 very quick questions, a curiosity more. Maybe I missed something. So they wait, the non-breeders, they wait, of course, to become a male. They cannot wait to become a female because of the size. This is the first one. The second is really a curiosity. How do they fight? How do anemonefish fight, and do they fight with the few that fight with the male or with the female?
Okay, so the first question first. The way the queuing works is that So the female is the dominant rank 1, the male is the rank 2, non-breeders are rank 3, 4, 5, and 6. So if the female dies, the male changes sex and becomes female,
Okay.
and the largest non-breeder becomes the breeding male, and everybody moves up 1 in the queue. So ultimately, there's the potential for every individual to be a non-breeder, a male, and a female in their lifetime. Does that answer that one?
Yes.
And then how do clownfish fight? So it turns out that the conflict is always most intense between the individuals most similar in size. And so under natural conditions, the only evictions I ever observed were at the bottom of the hierarchy, where these tiny rank 6 individuals or rank 5 individuals who are just 2 centimeters long would prevent new larval settlers from joining anemones and basically chase them around and kick them out. Okay, because what happens there is the smallest individual in the anemone has a size and it just isn't willing to tolerate a larval settler until it gets big enough. So under natural conditions, it all appears very harmonious because everybody regulates their growth to be 80% of the size of the individual above it because of this hidden threat of eviction. If you pair in the laboratory individuals who are similar in size, they'll fight and they'll fight hard. They'll chase each other, they'll nip each other's fins, they'll lock jaws, and we have to separate them if it gets too intense. Yeah.
Thank you.
Thanks.
Thank you, Eugenia.
Yes.
We go now to Siyabonga using GeneU's computer. Yes.
Hello, everyone. Thank you for the presentation. Siyabonga Sangweni from South Africa, currently working on the bush karoo rat. I'm just looking at the function of dry plant material that they feed on. And my question is based on the climate, the change in climate, in terms of what would be the implications of this, especially because it affects the coral reefs and their habitats. And as this decreases, how would this sort of have an implication on their group size or in their the change in their hierarchies?
Yeah, so that's a great question. Thanks, Siyabonga. I have a graduate student now, and Teresa has a graduate student, and this is one of their main interests is do the anemonefish confer resilience to the anemones to bleaching, and actually do the bleaching confer resilience to the anemonefish as well, by which we mean, does having a good group of anemonefish provide an anemone with enhanced resistance to bleaching and enhanced recovery from bleaching. So we had a marine heat wave in New Guinea this year. Temperatures got higher than we've ever seen them, over 5 degrees centigrade higher than normal. A very large fraction of the anemones bleached, and have then subsequently recovered. And what we're interested in is, do their anemone fish partners influence whether or not they bleached and how quickly they recovered? So we're in this sort of stage where the bleaching events are causing harm to the anemones, but not killing them. When the bleaching events are really severe, the anemones die, and then the fish are without a home, and then they get eaten. The relative to hard corals on the reefs, the anemones seem to be a little bit more resistant to bleaching. But as the temperatures go up, we're going to lose anemones as well. So.
Thank you.
Okay, thank you so much.
Thanks. I'd love to send a more positive message there, but.
Adriana.
Hi, everyone. Hi, Peter. Thank you for such a neat talk. I have a question from almost the beginning of the talk where you were basically explaining the experiment where you or your student moved the fish to a different anemone and then measure if they will come back.
Yeah.
And I didn't see a big difference between fish in the anemone with the female and with no female. Like in both cases, it was almost half of the experiments that they were coming back to their original anemone. So why is that? I mean, if you have a female already there, why don't you start breeding?
Yeah, I think that's a great question. And I think you have to think through what the experimental design was. These fish are living in an anemone. These anemones generally don't move. So they're in a stable anemone that's known, has a known history. They're associated with particular fish with whom they've resolved their conflicts. And now we are putting down an anemone on the reef next to them, which basically never happens. We're moving them to it. And so you can imagine that they could assess this as being sort of an ephemeral situation. This anemone has perhaps just been dislodged by a storm, which happens occasionally. They're with a fish with whom they haven't resolved conflicts, and it may just be better to move back and be in a healthy, stable anemone that you know you're going to inherit with some probability in the future, than sort of risking this sort of ephemeral anemone that we've just provided. So this is another reason why we think this sort of interaction with the anemone is now really interesting, and that there's maybe some benefit of being in sort of large, healthy anemones that we haven't yet been able to document.
Interesting. So that means that they have some sort of individual recognition of the members of the group somehow?
Yeah, I mean, I can tell them apart individually. The orange markings, the white bars, and the black are all very, very variable. So I can tell them apart individually, and that has all the sort of characteristics of an individual recognition signal as sort of described by Liz Tibbetts and James Dale in birds and wasps and things like this. So I'm quite sure that if I can tell them apart individually, they can tell each other apart individually. My best guess, and it is a guess as to what's going on, is that there's just some benefit of being associated with a large, healthy anemone that we have not yet been able to document.
Thank you.
Thanks so much. It's a great question.
Suleima.
Hi, Suleima Tang Martinez, animal behavior, retired from the University of Missouri in St. Louis. A wonderful talk, very interesting. I have a bunch of questions. I'll ask a few right now, and then I may come back later on for another one. I was wondering how groups form in the first place, and you mentioned that you could have an anemone fish settle on an anemone and then wait for years for somebody to show up to be a mate. And so I guess I have two questions. When that colonization event happens, is that individual that first settles a male or a female, and does it change sex depending on the size? Let's say it's a male. Does it then change sex to a female when the next individual comes in based on size differences? And how big do the groups get?
Great questions. Here's what I can tell you. So those events where anemones are defaunated, where there's no fish, are quite rare in the wild. I've seen a couple, and we've replicated that sort of experiment in the lab. And so in the lab, when we introduce, let's say we have an empty anemone and we introduce a size-matched pair of individuals, larvae at sort of 10 millimeters long, it's really interesting that they will actually enter into a growing race, much like non-breeding meerkats will enter into a growing race to achieve dominance. They'll both accelerate their growth, trying to outgrow each other, and then they'll settle down into a stable situation where you have one is the dominant who will ultimately become the female, and one is the subordinate who will ultimately become the male. The control groups in that situation, we have just one individual on its own, and you might think that it would grow just as fast as the rank one in the paired situation, but actually it doesn't. It seems to adopt the same strategy phenotypically as the loser in the paired experiment. And actually, we're showing its gene expression is also the same as the loser. And so what we think is that when an individual just arrives in an anemone on its own, it's basically in a sort of waiting mode to see who else turns up. Yeah. And if the one who turns up is a little bit smaller, then it's going to try and outgrow it and become the dominant. And if the one who turns up is a little bit bigger, then it'll let that one outgrow it and it'll become the subordinate.
Great.
So I have a PhD student, Lily Visor, who's sort of really interested in all these sort of mechanisms of how they're regulating their growth, either to accelerate or decelerate, depending on their position in the hierarchy. And then the second part of your question
was how big the groups get.
Right. So this is really fascinating. So because of this rule where every individual grows to be 80% of the size of its immediate dominant, anemone fish groups in the wild end up looking like sets of Russian dolls, where you have the female and the male is 80% of the size of her, and rank 3 is 80% of the size, rank 4 is 80% of the size. So it's just like a little set of Russian dolls. So the maximum group size, or the group size, is determined by the size of the female. And it turns out that her size is determined by the anemone size. And so in large anemones, you get large females with large groups. These dynamics are described in that 2006 Ecologia paper with Mike Kant. And essentially, we can show that you can perfectly predict maximum group size based on the size of the female and this 80% rule. So in the wild, the maximum size we see on a regular basis is 6 individuals, so the breeding pair and 4 non-breeders. And as a one-off temporary thing, I've seen a group of 7 individuals. So,
wow. So I have one more question. You mentioned that if larvae try to settle, then you have the smallest fish essentially evicted or chased it out. Yeah. Do you ever see competition among the non-breeders? And do you ever see coalitions forming between smaller fish to try to evict a larger fish that is trying to settle as a group?
So we've not observed coalitions in these groups at any time. It's very much a linear hierarchy without any sort of subgroups within the hierarchy. And I think that's because it's all relatively spatially constrained. So there's no possibility for a coalition to go off and develop somewhere. That was the question about coalitions. And the other question was?
I think just competition. Any competition among the non-breeders.
Yeah, I mean, I think the competition is just evident. It's evident from them restricting their size to be 80% of the size of each other. And so it's a really interesting situation, and where the group appears very harmonious. There's very little conflict under natural conditions because everybody regulates their growth to be 80% of the size. But if you tweak it experimentally, you get extraordinary levels of conflict. And this is all sort of ties in very nicely with Mike Kant's idea of sort of hidden threats being the sort of scaffold that underlies animal societies. Is, you know, everything can be very harmonious until you break the rules, and then there can be quite significant consequences. And it has really nice human parallels as well, right? I always think of driving on big open roads in the United States, and everybody is driving at 65 miles an hour, and you ask why they aren't driving at 80. And the reason is because there's a state trooper behind the next bridge, right? And it's this hidden threat that maintains cooperation within the societies.
Great, thanks.
Thank you.
So we have a question from our colleagues watching via YouTube, Lauren, and maybe after that you had a question. And then we'll go to Ana Maria Romero, who is a student. So let's take those 3 in that order.
Great. So this question comes from Shakti Lamba. It says, interesting work, Pete. Thanks. And the question is, what does the demography of your study population look like? Are clownfish found in many parts of the world? Could relatedness and migration patterns vary in different populations?
Yes. So depending on how you count, 28 or 30 species of clownfish, of anemonefishes that are found throughout the Indo-Pacific. The orange clownfish, Amphiprion percula, is found just in Melanesia, basically. You could imagine, for instance, that relatedness on very isolated islands might be very different from relatedness within groups on very continuous reef, right? So if you're on an isolated island that is over 100 kilometers from anywhere else, which is about the long distance dispersal of anemone fish, then you would imagine that island would build up relatedness within groups. But if you're on continuous reef where there's thousands of anemones, then you would imagine that relatedness wouldn't build up. So there's potentially interesting variation at the scale of the species. Species differ at the scale of different populations, and then at the scale of different groups within populations who might have different proximities to other groups, right? So all of these nested scales of variation could cause differences within groups. Thanks, that's a great question.
And I think you answered this, but the follow-up they had was, relatedly, do host anemone appear in clumps or with variable distribution patterns?
Yeah, so, I mean, it's really interesting. If there's anything I want to put a plug in for at this talk, it's that more people start studying anemone fishes, because there's so much interesting interspecific variation that's basically untouched. So there are populations of some anemone fishes that occur in the tropics and in temperate zones, and the ecological constraints in terms of vacant anemones and predator abundance seems to differ quite a lot between those regions, and then movement differs quite a lot, and then the whole group dynamics tends to differ. So I think there's some fascinating questions for people to explore in all the other anemone fish species.
Great, thank you. I had a couple of questions. I apologize if I missed this. I got distracted for a moment, but I want to come back to the question about tolerating the subordinates. Excuse me. And I think this follows up on the bleaching question. Do they produce some kind of mucus or some kind of chemical that helps to maintain the anemone itself, and therefore having more individuals around could enhance that interaction and maintain the quality of habitat they're living in?
Yeah, so while we haven't documented the effect of the fish on the anemone or the effect of the anemone on the fish in an experimental fashion, there's lots of anecdotal reasons why this hypothesis is plausible. So it's a protection mutualism. The fish protect the anemone from butterfly fishes that would eat it, from turtles that would eat it. They also, so that's been known for a long time. It's also known that now that it's a nutrient mutualism. And so the anemone uses waste products from the fish, nitrogen, phosphorus, to grow in these oligotrophic waters. And then what's really interesting is that, the anemone, a healthy large anemone, basically defines the foraging space for the fish. And so the bigger the anemone is, the more foraging the fish can do. The larger they can get, the more eggs they can produce. There's also transfer of nutrients more directly from the anemone to the anemone fish. The anemone fish will, the other context they'll fight other than eviction is in the context of fighting over anemone feces. And so, again, I think of sort of acacia ants and the acacia and these sort of little bundles of nutrients that they're provided with that seem to be being provided by the anemone in this case, and the fish will fight over them. And so there's a really interesting exchange of nutrients from the fish to the anemone and from the anemone to the fish that makes this hypothesis plausible. It's just not been tested experimentally. So.
Sure. And just a quick follow-up. Is it possible, or do you know, behaviorally, I might tolerate having a bunch of individuals around me if I don't have to deal with them. And are they sort of subdividing on the anemone in different regions, and therefore sort of just ignoring one another? So it's not...
Yeah, I mean, so one of the questions we haven't answered yet, but we actually one of the students is interested in doing it, looking at the videos we've taken through the bleaching event, is the sort of subdivision of space on the surface of the anemone. And we think there might be, I mean, it seems that the dominant patrols the whole area. I mean, these anemones are on average sort of 40 centimeters across. It's not a huge foraging area for a dominant, so they're probably patrolling the whole area. But we do think that subordinates sort of adopt sub areas within the anemone, perhaps to sort of reduce conflict in addition to being different in size, right? So there's like a size separation and a spatial separation. We just haven't, that hasn't been the focus of our studies till now, but it's certainly something that we could investigate now we have the videos. I think that question becomes more interesting in things like the cichlids, like Neolampologus and Sozenangs, bicolor angelfish, where there is more space. And so they can sort of get away from the dominance. And then you have this breakdown in the size hierarchies. So while we haven't done that and we don't see that in orange anemonefish, other people have some sort of interspecific evidence that when there's more space, the dominants have less control, the subordinates behave badly, and everything breaks down. So, yeah.
Thank you very much. I really enjoyed your talk. It's really fascinating.
Thanks so much.
So we'll take a question from Ana Maria Romero, then I will ask a question, and then we'll go to Cristina. Ana Maria.
Hi, everyone. My name is Ana Maria. I'm currently an undergraduate at Universidad de Rosario, working with Adriana Maldonado. And I have a few questions. First of all, what's related with how does the non-breeders actually get to be in the anemone? Like if their movement is restricted, but they're also not related genetically with the pair, how does that happen?
Right. So that's a cory fish ecology question. So almost all cory fishes have a bipartite life cycle that has a dispersing larval phase and then a reef resident phase post metamorphosis. And so in the case of clownfish, the eggs are laid next to the base of the anemone. The male primarily cares for them for about 7 days. Then they hatch and they disperse for about 7 days to 2 weeks. Okay, so these are larvae that are about 8 millimeters long. They go out into the ocean, they disperse some distance. And we actually know that distance now because people have tracked it using genetic parentage analysis. And so they can disperse anywhere from 0 to 120 kilometers. Okay, and then once they reach the end of their larval dispersal phase, they settle back to the reef, and then they locate suitable host anemones using chemical cues. And once they've found one, they'll make their best effort to enter that particular anemone. Okay, and they will be successful if the lowest rank resident is large enough to tolerate them. And if it's not large enough to tolerate them, they'll be unsuccessful. And then they'll probably perish.
Okay, and my other question is related with in rank 3, there can be multiple non-breeders. My question is if there is like a size difference between rank 3 or are they all equal? And this question goes to when rank 2 or male individual dies, what happens when there are multiple non-breeders? Like which one will be in rank 2? Right.
So I'm actually going to share my PowerPoint for a second because there's a good photo of what these groups look like. Well, this is a nice photo. If you can see this is the female. This is the rank 2. He's 80% of the size of her. This is the rank 3. This rank 3 is 80% of the size of the male. And this is the rank 4, who's 80% of the size of rank 3. So in a schematic sense, the groups look like this, right? And very much, so this is rank 1, rank 2, rank 3, rank 4. And this is sort of an unavoidable size hierarchy. If rank 4 tries to grow, it gets evicted. So it stays 80% of the size of rank 3. And then this is the order in which they inherit breeding positions as well. So if the male dies, the largest non-breeder inherits the male position, and this rank 4 inherits the rank 3 position, and they all move up one in the size hierarchy. Does that help?
Yes, yes. And like we've talked about how if rank 2 dies, all the other ranks will come up, but does it happen the same when the rank 1, when the female dies? It all happens, or the group dies? Like,
yeah, no, if the rank 1, if the queen dies, then the male actually increases in size to take the female position, and everybody else moves up following the male. Yeah.
okay, thank you.
Thanks so much. Those are great questions.
So I have 2 questions. The first one is non-scientific, very simple one. I was just checking that the movie Finding Nemo came out 20 years ago. And I just wanted you to comment on the extent to which you think, in the case of Nemo and that movie, what are the effects that you would say had a movie like that and the impact on society across the world, really, on behavioral ecology as a discipline, and more specifically on your research?
Well, on my research, the first paper, the Nature paper, came out right around the time of Finding Nemo. And that paper was all about growth regulation, and all anybody in the press could talk to me about was sex change, because I mentioned in one line sex change. But it did result in me having the #1 most emailed Yahoo news story globally for one day. So, as they say, no press is bad press. Even though I couldn't get people to talk to me about the growth regulation, they just wanted to know about sex change. And beyond that, I'm not sure how much of an impact Nemo has had on the world of behavioral ecology, other than whenever I talk to undergrad classes, it's something that is relatable to every single undergrad. Like every single undergrad for the last 20 years has known what a clownfish is. So that's certainly facilitated my talks. And it's always fun to sort of give them the brutal reality of what Nemo looks like, rather than the soft, fluffy Pixar version.
I said Disney, you're right. It was Pixar, right?
Well, it's Disney. It's Disney Pixar, right? So,
Disney
yeah,
Pixar.
same.
So I'm gonna move from one end of the continuum of science to non-science. We're gonna go from Nemo to, I need to ask you towards the beginning, and you know, Peter, that I'm taking notes on this because I'm teaching a course out of this. At one point, at the very beginning, you said that you went on to doing experiments because you couldn't infer causality, because you knew that there was some relationship, and that's where I lost you, that you knew that the size of the female was related to X, and this was related to Y, and because my perception, that's what I wanted you to clarify, is that because you knew about all the confounders, you went on to doing experiments. So probably not because this is unique to the system, but the system is such that allows you to identify and be clear and aware of all the confounders that require an experimental approach to get closer to causality. Most of us just go through life not knowing all the confounders and hope for the best. So did I understand well? And what were those factors that you knew you needed to control for?
So, yeah, I think in general, in this sort of experimental tests of cooperative breeding theory, it is not uncommon for breeder quality, whatever that means in inverted commas, territory quality, and group size all to be positively correlated. And because of that, it's been clear for some time that if you want to understand the effect of non-breeders on breeders, just taking an observational correlational approach doesn't really work. And so you have to do an experimental manipulation where you remove the non-breeders. In anemonefish, which is all tightly constrained within the space of the anemone, that effect is really, really clear, because the size of the female is positively related with the group size for all the reasons I've explained, because of the rules of the size hierarchy. group size is positively correlated with anemone size, and anemone size is positively correlated with the female size. And so all these things are closely related, and the only way to disentangle the effect of the non-breeders on the reproductive success of the breeders is to remove them.
Thank you. So it is, if I follow, it is what I'm saying. I mean, most of us have as many factors that correlate to the one of interest as you do, only
Right.
that I cannot remove the monkeys
Right.
from the forest. Right.
Yeah. This is the advantage. I mean, fish sort of fill this sweet spot, right, between social invertebrates and social mammals, in that they're vertebrates, but I can still do some experiments on them that people working on mammals can't do. So
I wonder, that could have been a negative effect of Finding Nemo, that suddenly people would be up in arms if you were to do any manipulation of our little fish.
yeah, I mean, over the years, I've been quite careful in sort of justifying the experiments that we've done, and I haven't, while one might anticipate that negative feedback from some quarters, I've not experienced it personally. So
Thank you very much. So let's go to Cristina first, then we go to Gabriela Peña Ovelio, and then, no, I'm sorry, Laura Mercade, and then to Adriana. Cristina.
So thank you, Peter.
Thanks,
Wonderful talk, wonderful model system. I'm based in France, Sorbonne, Paris Nord. I work on hermaphrodites. I've worked on social insects before. My question is, in a way, a little bit technical. You were talking about non-breeders as non-active, non-reproductively active individuals. And my question is, are they sexually mature as males, or are they really immature individuals?
They do not have functional gonads. So,
okay.
So
no way to act as sneakers or something like this?
No. So Fricker did the gonad histology in the 70s and showed that they don't have functional gonads. Because of the interest in dispersal, people have done a whole bunch of parentage analyses, and they've never traced babies back to the quote unquote non-breeders. So they truly are non-reproductives. And I think one of the nicest experiments to show why is that experiment of Teresa, when she introduced a breeding male of the same size and it just gets evicted, right? So if they turn on their gonads, they're going to get evicted. So they keep them turned off. Yeah.
Okay. Thank you.
And I think that's why, because I think the threat of eviction is why you don't see sneakers in this system, actually, because it's so easy for the breeders to evict, given the confined space and the power differential, that I just don't think, it's not like a sneaker can come in and then run away somewhere else. It's like, if it sneaks, it's going to get killed. So.
Thank you.
Laura, you're next.
Hi, I'm Laura. I am a research assistant at Leiden University, where we work with great apes in comparative cognition. And I have one question, maybe it's a bit tangential to your story, but I was wondering if you know how the fish know that there are 80% of the fish above them.
I wish I knew. Yeah, I mean, it's a really interesting problem, right? Because they grow to 80% of the size of their immediate dominant to within 0.1 of a millimeter. And so both individuals have to know their size and their relative size, and how they do it. We have only just started to touch on. So I had a master's student, and she sort of paired individuals to see if they would engage in this growing race. And she put different kinds of barriers between them, such that they only had olfactory cues, or they only had visual cues, but not olfactory. And she showed that actually any barrier interfered with the process. And so it really seems like they have to physically interact with each other it could be that they're swimming side by side and sort of visually estimating their relative size. Fish also have a really sort of stereotypical submission signal, which is if a dominant chases a subordinate sort of lies at an angle and quivers from head to tail like this. And I think it's really interesting that that could potentially be sending a pressure signal that's detected via the lateral line. And so fish have this extra sense, which is the lateral line, which detects pressure waves. And so basically they could be generating a pressure type signal that is an honest signal of how big they are. That's all hypothetical. We haven't done the critical experiments to figure out how the sort of communication aspect of how they're regulating their size. I have a PhD student now who's working on the sort of gene expression aspects of how the subordinates regulate their size, understanding at the genetic level what they're turning on and off to turn their growth on and off. But the sensory communication aspects we still haven't really grappled with. Great question.
Thank you.
Not tangential at all. It's a great question.
I have another, Laura, if it is okay with you, I'll ask you a question that relates to Peter's work, but you're the great apes. I mean, I meant to ask this to Tony and I should probably know the answer, but I don't. And since you're sharing with us that you work with great apes, Peter, in your paper on social regulation of growth, there's a section on mammals and humans, and you make reference to the orangutans. And I meant to check on this, I didn't. I mean, for those of you who are not aware, we've known for a long time that male orangutans, some of them do not develop full growth. And at least up until 20 years ago, which are some of the references in your paper, that was what we knew. And I think that, do you know, Laura, where we're with orangutans, if it is true that those unflanged males really grow fast, or if it is more like it's an alternative reproductive phenotype where they stay small and reproduce? Because I don't think they show the kind of social growth changes that we've been talking for other taxa. Do you know, Laura?
I have to say, Eduardo, I have this job for 1 month and I haven't worked with orangutans before that, so
I should know as well. Don't worry. But I thought maybe you know it. I need to check it because I don't know where the orangutan people, of course, these guys, I mean, they take 50 years to grow, right? So go figure out what is fast for an orangutan to grow. But I need to go check on that. Anyway.
We ran that paragraph by Cheryl Knott here at BU, who works on orangutans. It was, she didn't throw it out of the paper.
And that paper is that's a recent paper, right? So if Cheryl says yes, then that's recent. I don't remember the date of the paper, but it's in the last few years.
No, it's a 2022 review.
Oh, okay. No, that's good to know. Right. Oh, there we have the answer then. Yeah, great.
So I think that's robust, and the human data on sort of social regulation of growth in humans is more sort of anecdotal, but I think interesting to pursue. So we're talking about this is the fourth in the list of the papers that was given. It's a review that we did with Tim Clutton-Brock on social regulation of growth or strategic growth in fishes and primates, in vertebrates, social vertebrates in general. So if anybody's interested in that, there's also a paragraph on trees, if anybody's interested. But
thank you. Adriana, I think you're next.
I think we're right at the right spot for my question, but I forgot to introduce myself last time. I'm a professor at the Universidad de Rosario in Colombia and a co-organizer of the PINE seminar. And my question relates to how groups are, the composition of the groups in relation to the size of individuals. So we've been mentioning once and like over and over that they are like, there is this rule of the 80%. But in one of your slides, you showed also like a distribution of proportions of different sizes. And if I remember that correctly, there is like at the very end of the tail, I think proportions are similar, which if I understand that correctly, it means that individuals within that group are more similar to each other. So it doesn't really hold for the 80% rule, or did I understand that correctly?
I think this is the figure you're referring to. This is the
Yeah.
distribution of size differences occurring under natural conditions. And you can see that there are a few individuals within groups that are similar in size under natural conditions.
Yeah.
These would almost always be at the bottom of a hierarchy where you have two individuals engaged in conflict. And so this sort of pointed group entry. So there's probably, I forget exactly how many pairs are in here, but it's not that many. And I can almost guarantee that they are sort of the 5th and 6th ranked individual within a group, or the 4th and 5th ranked individual within a group who are in conflict that hasn't been resolved.
Oh, I see. Okay,
but you
so
also,
these are, go ahead.
No, go
on. So these are, we could say that those are transient groups, like in an unstable state somehow?
Yeah, the majority of the sort of top end of the hierarchy is probably stable. But at the bottom end of the hierarchy, there can be unresolved conflicts under natural conditions. And so that's often where you see sort of let's say the 5th ranked individual is 20 millimeters long, and then a larval settler turns up who's not dissimilar in size. And so they are in this conflict that can take a while to resolve. And it can resolve in one of two ways. Either the slightly larger individual can grow big enough that it ends up tolerating the subordinate, or that the larger individual can't grow because there's no space above it to grow, and it ends up evicting the subordinate. And there's some really interesting strategies that occur down here. So individuals that have just settled into groups and are receiving aggression, they don't develop these bright orange, black and white colors. They remain sort of pinkish and camouflaged. And it looks like a strategy to sort of avoid eviction when they're in conflict with these other individuals. So that's cool. That's what's going on here. But you also point out quite rightly that I'm talking about an 80% rule, but I'm focused on like the mean, right? There's lots of variation around here that is potentially biologically interesting, that one might ask, why are individuals having smaller size differences in some groups than others? What effect does that have on conflict, and what effect does it have on group productivity? That these are questions we haven't explored so much, but you're right. For simplification, I talk about an 80% rule, but there's lots of variation around that. That's interesting.
Yeah, that's pretty interesting. And the effects of that, like in fitness, or how the groups works as a collective, that's opens new questions. Thank you, Peter.
Yeah, I mean, actually, I'm going to follow up there, because one really interesting, there's another anemone fish which uses the same species of anemone in the same lagoons in New Guinea. It's called the pink anemonefish. But it has a few really interesting differences, which is that the non-breeders seem a bit more mobile and a bit harder to evict. And this has a multiple knock-on effects, we think. First of all, it means that if a vacancy opens up nearby, they can move and take that vacancy. Because of that, it seems like subordinates in these groups are less willing to remain small because they can have their inheritance usurped. Because of that, those groups end up with more conflict. And so you have two species of anemone fish occupying the same anemones in the same place, but this difference in one biological trait in how mobile they are seems to have multiple knock-on effects on the society in terms of conflict. And those groups are really interesting because occasionally it seems like the groups get so big and so full of conflict that the dominant female gives up. She leaves, she swims down the reef, she finds a group of orange clownfish, she kills everybody, and she starts from scratch on her own. It's really cool.
And she lives on her own. She doesn't take anyone with her.
Yeah, it's almost like she just gives up because of the amount of conflict amongst her subordinates. She takes off, she goes to a new anemone, kicks out all the orange clownfish, and just waits for somebody else to move in with her.
Interesting.
I want to give
Again,
Thanks.
really interesting interspecific differences that haven't really been quantified or explored. So
Yeah.
I'd
Great.
like to take a break now, and Peter has some meetings with the organizers of the fine seminars and our students at 1. So I think it's fair that we just give you at least 15-minute break. With this, I'm going to stop our live streaming. Thanks to all of you who may have joined us